So we just talked about the different little machines and I gave you the names of them. And now I'm going to walk you through exactly how those little machines work to duplicate our DNA. So the first step is our helicase splits apart the DNA. And then our single-strand binding proteins keep the DNA from snapping onto itself while we're waiting for the rest of the machinery. But what is the rest of the machinery? Well, we have polymerase, and we have primase. Those are the main ones. We also have, and then we have DNA polymerase 1. So we have DNA polymerase 3 up here and polymerase 1 down here. And then we have DNA ligase. Okay, and now let's talk about how these 4 little proteins work together in concert to replicate all of your DNA, which this is fascinating. Okay, so let's assume out here, and I'm going to draw it so it's straight, but you need to remember that the DNA is not straight, it is wound up, which is why you need helicase, right? DNA is all wound up, but I'm going to draw it straight because it's easier. And so, what we have is out here, after our DNA polymerase 3, I'm going to draw this very specially, a little purple section, and that's the start of our sequence. Now, this purple sequence had to be put on by the DNA primase. It primes the DNA. Now, that was put on by the primase because the polymerase 3, and this is polymerase 3 down here, polymerase 3 will not act on a single strand of DNA. It needs a double strand and only a little bit, 10, 20 nucleotides, and then it can get started and do its thing. And so it started here, so the primase put on, I've got it backwards. All right, so the original strand of DNA is all black. Our new strand has a primer. And that new primer was enough double-stranded length in order for our polymerase 3 to bind and move along. Okay? Very straightforward for that strand. As a note, after the DNA was separated into 2 single strands by the helicase, single-stranded binding proteins stuck on there to keep it from attaching. That's kind of a thing that people tend to miss in these early classes, and I think it's just there's no reason for you to not know it. It's not that hard. Okay, now let's talk about the lagging strand. So, and I messed this up, but I will try and remember it. So our polymerase moves from 5' to 3'. Let me check that. Okay, so the polymerase 3, to be specific, works in the 5' to 3' direction. And it's important to note that that 5' to 3' direction is in the context of the new strand. Right? So this here is the 5' point of the new strand, and this is the 3' point of the new strand. Now, what does that mean? What does that mean for our lagging strand? Well, remember, this DNA up here, and say it with me, antiparallel, it is anti-parallel. Remember the term anti-parallel. Or do your best to, write it down or something, I don't know. Come up with a mnemonic if you can. I love mnemonics. Okay, so it goes 5' to 3'. And then it can duplicate all the way through because it's going in the same direction as the helicase. But what about in the case of the lagging strand? Now, as we will learn more about this process, and I'll get onto a proper series on DNA replication later on, but it's kind of magical the way this is done in terms of the polymerase 3 is actually a macromolecular complex, and it's pretty fantastic the way it works. It actually has 3 polymerases, and there's one for the leading strand, and there's actually 2 for the lagging strand. They kind of swap over like a pit crew. So there's always one of those lagging polymerases that's working on the lagging strand. Not important right now. What is important right now is that this direction doesn't work super well. So let's talk about that. So you're here, and I've drawn it very intentionally like this, and this is called a trombone configuration or trombone replication. And the reason that is because our DNA replication machinery, our polymerase, has to go away from the replication fork. And so this trombone section, and as we'll begin to see, it kind of makes sense, but it gets real big and then it shrinks. And I'll explain how it gets real big and shrinks in just a minute. But first, we'll do an example just out here. So my primase, now because we're going that way with our polymerase, my primase puts on a primer here, and I'll draw the example such that there's another primase elsewhere. My primase puts on a primer here, and it goes this way towards another primer that's already been put on. Understanding that we have to go away from the replication fork because of the directionality of polymerase. So now, what this trombone-like structure does is it stretches out, and when we get the chance to put on our primer at this point, because we only have so many polymerases and they need to be kept close, so once we've gotten all the way out here and we've finished our replication, we go back up here and we start again. And so, because we're going away from the replication fork, we can kind of eat it up. So, we put on a new primer just behind the replication fork, up here, and then our polymerase kind of eats it up. And this configuration would change, it would just kind of get spat out the back. Okay, but now we have a small problem, because the polymerase is not able to complete the last bond between the nucleotides on these primers, with the new strand of DNA. And so in order to achieve that, we need 2 things. So we have still these primers, which are not proper DNA, and so in order to get rid of those, we need DNA polymerase 1. And DNA polymerase 1 removes those. It removes just the primers. It'll remove like maybe a little bit extra either side, but the important thing is that it gets out the primers. And now you have a little canvas where your DNA ligase can fix the gap. So the DNA polymerase 1 removes the primers and replaces it with DNA. And it's important to note that these primers are RNA, not DNA. They're RNA, not DNA. Which means that we can't have them in our genetic code. They need to be replaced. So that's why we replace them. And finally, our DNA ligase just plugs up the little one-ended gaps. And that's how it all works. And now I think I might go back and talk about a replication bubble, because I get it better now. And I think that's useful. So we have our double-stranded DNA. Right? And we go one direction and the other direction. So we've got our helicases, helicase B's. And in each direction we're helicasing in each direction, and it's splitting it up. And we started our replication here, which means that we actually have a leading strand and a lagging strand on both sides of the bubble, right? So we have our primase come in here, and then our DNA polymerase goes nuts all the way down on that one. And the same happens on this side, because these are the two leading strands in our replication bubble. Right? But then in our lagging strands, of course, we couldn't just go straight for it because we can't go in the correct direction. So we need to add multiple primers, and then we need to fix it up as we go through. So we're splitting apart in these two directions, but this guy, right, can only replicate in this direction. So this section, which is about to get shown, can't have a primer behind it, can't have a primer up here. So we need to add a primer as we go. So we use those SSBs to keep it from reattaching. We add in a primer here. We go as far as we can with our DNA polymerase. We put in another primer. DNA polymerase goes as far as you can, and etc. Until we're back to the origin of replication. Now, as we recall, the DNA polymerase 1. So the DNA polymerase 3 does the bulk of the work. DNA polymerase 1 removes the primers and replaces it with DNA. And then finally, DNA ligase fills the gaps. And then we have our complete, nice, new double strand of DNA. Thanks, guys.